Good morning and welcome to our next session, our second session of the day, entitled Navigating the Drug Development Roadmap. So as many of you might know, Global Genes has developed a research roadmap, a copy of which you will find in your program. And this particular session will really guide advocates on the critical stages of drug development and how advocates can effectively engage at every step of these stages. I'd like you to all please welcome our panelists, Suki Bagal, Liz Buttermore, and Ebony Dashiell-Aje.
Oh, yeah, we'll give it a minute till folks gather in the room. I would like to have the roadmap on the screen, please. Yeah, perfect. Yeah. One more minute. Good morning, everybody. My name is Suki Bagal, and I'm honored to be here today to kick off this discussion with two of my panelists, whom I would actually request that they introduce themselves, because I just like it when people tell their own introduction and their own story. So Liz, may I just request you to go first?
Yeah, absolutely. Hi, I'm Liz Buttermore. I'm the Director of Translational and Vitro Models here in the Translational Neuroscience Center at Boston Children's Hospital. So just to kind of give a flavor for who I am and why I am here and how I got to this point, I can start by saying I've always been somebody interested in the pursuit of new knowledge. I was fascinated by the ability to be the first person to find something out. That led me to a PhD in developmental neurobiology and kept me excited to be in the dark confocal room for hours and hours on end, because I was going to be the first one to know, if we remove this gene, how does that affect the organization of myelinated axons in development? And I just thought that is so cool. When I finished my PhD, I wanted to shift to something more translational that would be able to have more impact on patients and drug development. So I actually came to Boston Children's to complete my postdoc in Clifford Wolf's lab, working on developing models for painful neuropathies. Here, I learned that I didn't need to take images one at a time and count by hand the synapses or neurites, but there are actually programs that can automate all of those processes. So now I can simultaneously image thousands of conditions and quantify them automatically. And this really changed the questions that I could think about asking. After my postdoc, I joined the Translational Neuroscience Center in the Human Neuron Core. We're using induced pluripotent stem cell models to model neurodevelopmental disorders. Induced pluripotent stem cells, for those who aren't aware, are stem cells that derive from skin or blood of patients with a particular disease. So it really provides an opportunity to study that patient's cells in a dish. So my group in the Human Neuron Core is developing tools and assays and endpoints for neurodevelopmental disorders. And then I also lead a biorepository core for neurological samples. So this involves collecting CSF and brain tissue from patients with neurological disorders for research purposes. And so I'm excited to be here today to tell you about my work and kind of how that fits into the roadmap here.
Ebony.
Hello, everyone. Such a pleasure to see you guys' great faces in the audience. I'm Ebony Dashiell-Aje. I currently head the Patient-Centered Outcomes Science Group at BioMarin, and our primary goal is to drive the research strategy, including endpoint strategy, on how to measure how patients feel, function, survive in clinical trials and other aspects of interventional and non-interventional research. And prior to BioMarin, I actually was a regulator. I worked at FDA. Not sure if there are any of my friends in the audience, but that was a privilege to be able to serve in that public health capacity. It was quite impactful, specifically because I was able to contribute to the patient-focused drug development guidance series that a lot of researchers in the audience probably use to define exactly how they should approach collecting patient experience data. And before that, I was a consultant. I worked at Evadera, a CRO, to be able to generate evidence for multiple sponsors. So I've been in health outcomes research for quite a while. But I think, my passion for this work is because I care about bringing the most transformative treatments and therapeutic options to people who need it most. And I value, the elevating the patient voice and caregiver voice in research is critical to bring forward, efficient solutions where we can bring treatments to market the soonest to those who need them. So, and I also play a role as a great, rare caregiver and advocate. My mom, she has a rare disease, cavernous angiomas. My daughter had leukemia during the pandemic, guys. Thank God she's in remission, but she had leukemia. And then my son was recently diagnosed with type 1 diabetes. So as a researcher, while my work is centered around robust evidence, I also have the vantage point of knowing what it takes to really drive this field forward. So it's a privilege to talk to you today.
Wow. I'm just amazed at the expertise that we have between the two of you. And I know you both have very enriched experiences. And for the audience, we are going to break that down to make sure you understand how it all translates to patients and patient advocates such that they can be on a successful path of the journey for drug development that they're seeking. So if I may just ask the audience to take a look at this roadmap. Take a minute, just find a starting point and then try and reach the end point and just focus on some areas where you think as a patient or a patient advocate, you can contribute, and just keep that in your mind. You don't have to say it, write it, or express it. Just in your mind, keep that. And through this panel discussion, we will try and align on points where patient advocates can align with certain steps of the journey to make sure that we have, the path that doesn't look so linear, at least has a pattern that can be feasibly followed. So please just take that minute now, and then we will start a discussion. And I think there's a copy in your package in your bag, so please use that as a guide as well. Just to kick off our discussion, Liz, I know you gave a great introduction for your journey, for the work you do. Can you just explain to me how your role, how your work, how your career path fits into this roadmap for drug development? What are the points of convergence? If you can just briefly explain that, that'll be helpful.
Yeah, thanks. So in the human neuron core, as I said, we're working on developing tools in the preclinical pipeline. So that would be the first stages of this roadmap, including basic research, understanding the diseases, developing this research toolbox. In my case, cell lines, iPSCs, the biorepository of patient samples, completing the preclinical research leading to drug discovery and screening. And I think, the key for us is really wanting to make tools that are available for the research community. And so I think that's one of the places when foundations get involved in helping to establish these tools, these IPS lines, we find they're really more widely shared across the research community so they can be used by more researchers. So we're interested in taking these stem cell lines, identifying differences in the patient and control neurons, and then leveraging those differences to screen for compounds that reverse the difference, make the patient neurons more similar to the control neurons. And when we have these assays established, also wanting to share that information, that data with the research community to kind of lower the bar for other researchers to join in. And so again, I think that's where advocating and having the community and partnerships really lowers the barrier for more people to get involved in the same projects.
How about you, Ebony? Can you just share some points of convergence with the patient centricity of the work that you do in the landscape?
Yeah. So believe it or not, what I do is supposed to contribute end to end, right? The role that I play is making sure that the patient voice is elevated in a robust way, like turning insights into data and convincing all of the cross-functional teams across the medical product lifecycle that it's important to consider what the patients want to measure most in a trial at the earliest stages. And this can go all the way to the preclinical and early development stages. And the discovery phase is also important too, because, the patient and caregiver voice can help guide, what are the most, relevant unmet needs? How can, a specific asset or candidate tie back to those needs and creating a treatment that will actually improve something? Also, when it comes to endpoint selection, deriving endpoints, picking the different measures that are relevant to incorporate in a trial once we get to clinic, those types of exercises, the research behind it, quantitative and qualitative research, all of those things are relevant for what I do. And also thinking into the future and trying to promote access of our treatments to patients that need it most. There are later stages of the drug development process, even post-market, where the work that researchers like myself, methodologists, measurement scientists, we contribute to even maintaining that robustness of evidence generation after the trials are done. And that's really super cool to me, because I remember back in the day when we used to think that patient insights were only relevant for clinical trials. But now I think, there's enough momentum, both in the community as well, the research community, as well as regulators and payers, to emphasize the totality of evidence and how multiple studies, not one study, will serve as the basis of proving that a treatment is actually beneficial to someone. And it's important to have experts who are there to guide the measurement, because just a questionnaire is not necessarily reliable and valid, or, measuring things that they should be measuring in a trial. So that's really the context of the work that we do, how it's end to end from the, what did I say? Drug discovery, preclinical, all the way through to post-marketing, where the work that we do touches every aspect of development.
If I could just build on that too, I think one thing Ebony said that reminded me something that's really important, even in the preclinical stages, is it helps us to know what is important to patients and patient families, what is meaningful improvement to you. Because once we know that, we can make sure that whatever model system we're building or assay or biological question we're asking provides an opportunity to identify drugs that address that impact. So for example, for a neurodevelopmental disorder, a patient group might say that improvement in seizure burden would be a meaningful improvement to them. And so in that case, we can develop an assay where neuron hyperexcitability is the readout. And if we can reverse that hyperexcitability, maybe we can control seizures. Others may say that regulating sleep is more important, or other parts of the disease. And so having that information early as we're developing the assays, as we're, before we run screens, is really important to make sure we're meeting the patient needs.
Can I say one more thing about partnership and collaboration? So whenever we are specialists, we oftentimes focus on what we do best and we work in silos. But one thing that I can say as my career has progressed, I've seen more collaboration across multiple business units, multiple departments, as well as entities, between the patient community, patients, caregivers, researchers, all coming together to try to answer the biggest questions earliest in the process, not waiting until you're getting into clinic to start thinking about, well, is this really something that would be impactful for patients? And I value the close collaboration that we started to develop with our early research colleagues, them bringing us into their ideation processes and strategic discussions, and even the dialogues that they're having with academic institutions and labs, so that we can provide, some guidance on how best to bring in the patients and caregivers to give input at the earliest stages, and even collaboration with advocacy functions. So our strong advocacy department provides that relationship that we can then leverage to, outreach and communicate with folks who could best provide insights for our teams. So I just think that now that we're evolving in data generation, we should really focus on that, collaboration and being creative about when we can leverage information the best.
Wow. So clearly the patients are at the center of all this research being done. If I were a patient, I would have some simple questions. Perhaps, Liz, you can explain to me, what is a biomarker? Because again, people from the science, they probably know it. Actually, in my experience, most people don't know it, but they don't admit to it. So just please throw some light on what are biomarkers, because I think that'll go a long way in kind of bridging the knowledge gap.
Yeah, absolutely. So biomarkers can mean different things across this pipeline, but in general, the definition of a biomarker is something that is measurable indication of a biological state or a condition. So if you think about this roadmap, it can be a molecule, it can be a gene, it can be a physical attribute. It can be so many different things that can relate to diagnosis. It can relate to the progression of the disease. So biomarkers for progression might be different than a biomarker for diagnosis. And then it can also relate to intervention. So how well is somebody responding to an intervention? The key is that it's measurable, quantitative. And so an example for Alzheimer's, phospho-tau levels in the cerebral spinal fluid go up. When we think about using biomarkers for research, we can look at CSF levels in different patient samples, but we can also think about, can we look at phospho-tau levels in our iPS-derived neuron models and develop a screen that addresses that pathway, those mechanisms? So biomarkers that can be translational from preclinical research all the way to the clinic are going to be really powerful for maybe improving confidence that whatever you're developing might have efficacy in the clinic and that you'll be able to read out that target.
And obviously for biomarkers, you need biospecimens, right? So can you just tell us the role of biospecimens and what role patients can play in making sure there is a biospecimen repository or access for biospecimens to researchers and vice versa? Because I think what we're trying to achieve here is connect the dots and reduce the gap between what's out there, what needs to be out there. So if you can just explain where the patient centricity can assist in having a robust biorepository of biospecimens.
Yeah, so biospecimen can be any biofluid, tissue, urine, blood, brain tissue, CSF, skin, any biopsy from any area, really any biological fluid or sample. And they're so important for research. both as part of natural history studies, which maybe Ebony can touch more on, but also for basic research into the biology of the disease. So in the repository that I lead, there's often new questions that we could not have envisioned asking 5 years ago. Groups at BCH looking at somatic changes in the brain. You can only ask that with brain tissue. Are there genetic changes only in the cells in the brain that are not seen elsewhere in the body? And those are driving disease. And so we can't, ask those questions unless we have access to that tissue. So creating repositories and having that tissue available, then when the scientists think of the next question, they can access that tissue and have it be more readily available rather than going back, recruiting patients. It kind of reduces, again, that barrier, that timeline to asking and moving the science forward.
So I think the point here is that research can only progress if there are biospecimens to be worked on, or labs can have access to biospecimens. So I think patients are the only source of those biospecimens. And sometimes patients don't realize the power or the importance or the relevance of their willingness to provide biospecimens is. And I think this is something that I would just like to highlight for patients and patient advocates to consider creating a community that is supportive of providing the biospecimens required for research. Because again, there can be multiple models, that can be multiple theories, there can be multiple chemical compounds, but really, unless you have the biospecimen to isolate a biomarker or study the effects of those molecules, there is no therapy. So I think biomarkers and biospecimens are a step in the path that we need to be on for drug development. So coming to patient data, Ebony, can you just share what is the relevance of patient-centric data and how it plays a role to link drug development to actually drugs coming to the market?
So it's very important to have the most appropriate tools that reflect how patients feel, function, and survive. A lot of times in rare disease drug development, all we have is surrogate endpoints, biomarkers, where, we understand the physiological response, but we don't quite know if that response actually equates to somebody's life being improved by a treatment. So that's where these measures, patient-centered outcomes measures, come in, because you need to be able to link those changes, those physiological changes, those observable changes from those lab values to, is this person feeling better? And the only way to know if somebody is feeling better is by doing what? You ask them, right? So that's where my career comes in. We develop the measures that can reliably and validly assess how a person is actually doing in their life as it relates to the specific concern that they may be trying to address with the treatment. And so when we're thinking about how this relates to drug development, it's super critical to not leave out that patient-centric measurement approach. A lot of times there's focus on how can we get quickest to clinic, and we ignore the fact that, oh, well, we also have to measure how a patient is feeling, functioning, or surviving. And when it comes to how to measure this, you measure that through what we call clinical outcome assessments. That can be different types of questionnaires designed to measure how patients are reporting how they're feeling, how their symptoms or the impact of their symptoms has changed during the course of treatment. You can also get reports from caregivers who are observing how their family member is also reacting to the treatment. Some things need to be a little bit more objective. You need to have some standardized, perhaps, performance outcomes, which can measure functioning in a clinic setting that's systematically measured across different contexts. But also we're evolving into more passive data collection through digital health technologies. How many of you all are wearing a smartwatch, what we call a smartwatch back in the day? Yes, I am too. I have one right here. And, you know, these types of measures really have revolutionized data collection, and they can be very patient-centric. So when we're thinking about biomarker data, the criticality of having these types of tools included in any research study really makes the difference for being able to translate how this physiological response ultimately means, you know, the best benefit for a patient's life.
So if I may just further that question, how do biomarkers, how does the translational medicine and patient-centric data, how does it help in optimization or selecting the best treatment that patient may have? In the sense, in the lab, there are multiple molecules that you're looking at. When you're collecting patient data, you're looking at different symptomatology. So what is the role for both translational medicine and for patient-centric data in optimization of selecting the right treatment or picking the right candidate to move forward? Because patients need treatments fast, right? And if you have 3 options, how do you pick the best one to go forward? So if you can just throw some light on that, it'll be helpful, because I think it'll help tie the relevance of collecting clinical data and the patient endpoints to how it together comes in as a factor which affects the selection of the candidates for treatment to be taken forward.
Sure. Yeah. So biomarkers as a starting point can be used for patient stratification. So whether that's a genotype that a specific gene therapy is amenable for, one thing maybe we'll discuss in a bit is, not one size doesn't fit all, even within the same disorder. So being able to stratify patients that are going to respond to a particular therapeutic strategy at the onset of the clinical trial will really lead to more successful outcomes.
In terms of candidate selection, at the earliest stages, engaging patients and caregivers, understanding what's most important to measure, that can be critical for deciding on go, no-go decisions in drug development. If you're understanding that your candidate is unable to deliver on what is actually most meaningful to the patients, then that can be a critical moment from a research perspective, drug development perspective, on how to change course, how to modify things, how to better design, in-clinic studies so that the design can reflect or be optimized in order to be able to establish treatment benefit. And so that's how it's sort of linked between the biomarker and the clinical outcome assessment. But I would say that as you move into clinic, it's even more critical to show the correlation, right? Because oftentimes the primary endpoint in a clinical trial is a biomarker. And so the relationship between the two is like that magic that convinces regulators, it convinces payers, it convinces the medical community and patients themselves that, okay, I can be confident in these results to know that it's truly measuring something that matters to me, and these results mean that it could benefit me potentially. So that's sort of the time.
So I guess the strong tie for taking a program forward lies in the quality of data. So can you just comment on what is considered as good data? Because data collection can be a daunting task, an expensive task, and people kind of can have various sources of really finding ways of collecting data. But can you just throw some light on what is considered good data that can really bring meaningful impact to drug development? Because I think data in and data out process is now passé, right? It's gone. We need a strategic thought process of what needs to be collected. So can you just explain what good data looks like for drug development?
Good data looks like data that is collected in a reliable way, that's collected systematically over the course of a study, and also that yields results that are deemed valid and reliable for use. And what does that all mean, right? Because that's all technical, data science, woo-woo. When you think about collecting data, there are principles, best practices to guide whether or not you've collected data in the most appropriate way. And you have to approach research from a research objective perspective, and then deciding systematically how best to achieve those objectives with the different elements of the study. That can be what type of instruments you're using to measure. That can be how long you're collecting data, who you're collecting the data from. When you're thinking about the principles to guide your research practice, there are a lot of guidelines out there now. So prime example, with the regulatory landscape shifting, there's been an evolution of really robust guidances through the FDA, Ph.D. guidance series that walks you through how best to select a sample that you're trying to collect data from. What should you be assessing in this sample? How is this sample reflective of the target population that you're trying to treat? Then exactly what are we trying to really measure in this study? What's feasible? Like, you can't boil the ocean. You have to be targeted in your strategy about what you can actually assess within the context of one given study and what's most impactful, right? And then you have to select, okay, well, how am I going to measure this stuff? So I know what I'm supposed to measure, but how am I going to do it? And there are questionnaires, there are labs, there are, different clinical markers and assays and things that you can use, all that come together in a study design. And then you have to focus on implementation. I think that's where people miss out the most. You have a lot of experts who might come together and have great ideas, but is it feasible to implement? Or how can you mitigate issues with data collection during a study? The implementation planning and strategic components of that, I think, are critical to data quality. If you don't have a way that you're systematically collecting data across studies, within studies, across sites, then you're going to introduce some error in your measurement that ultimately will prevent you from seeing what a true result is. And so I would say that going to the different guidances, there are also different best practices papers by multiple organizations like ISP or ISAQOL that can really help to illuminate all of the different steps. There's even COA roadmaps. There's different roadmaps that have been created by patient advocacy organizations, as well as regulators and methodologists. And those help walk through step by step, what are the requirements? One thing I can say is that with all these guidances, it sometimes is very overwhelming. It's overwhelming because you don't have all the money, you don't have all the time, and you're like, well, what does good really look like? And that's where that collaboration piece comes in, alignment with multiple stakeholders on, well, what is the best thing that we can do in order to maintain the integrity of our data? And that's on a case-by-case, study-by-study, indication-by-indication basis. But it is required for you to do some trade-offs, because you're never going to design the perfect holistic study. So I guess the key takeaway is that there are guidelines and guidances that can be used, that we don't want perfect being the enemy of the good, but collaboration is how you determine what is the best to the stakeholder that you're trying to generate data for, whether regulators, payers, medical professionals, or patients themselves.
Thank you. So if I may just ask, all these steps obviously involve money. What are the typical sources of funding that you both know exist and where there is room to grow in for both of your fields. So if I may just throw it back at both of you to address that, that'll be helpful.
Yeah, I mean, I think the challenging thing is that when you apply for NIH funding, they expect you to already have the tools, the IPS lines in hand. And so we've really relied on patient advocacy groups and foundations to provide the funding needed to establish these tools. And it actually, I think, provides an opportunity when the funding is coming from those sources, because there's not then a conversation about who these lines belong to, but they're made for the research community. And so it's easier to share them. They're made with the purpose for sharing. And so it's easier to distribute them to more labs. So I've found that getting the funding to create these tools and then using that to leverage for other funding sources like the NIH or partnerships with companies or different avenues after that.
And for the types of tools that we develop, we have to go through a validation process for all of the different types of measures, which is both qualitative and quantitative, and it's multi-year. And a lot of times, you have to figure out, well, what is the highest priority yield? And creating these tools, how best can we design something that's usable across multiple stakeholders? And we have, of course, sponsor funded work that we do, but I've found that the most progress is made by collaboration, so you have public-private partnerships, you have different, entities that come together, whether it be nonprofits, sponsors, research incubators, academic institutions that sort of pool their money together. Because I think that you can't do it all by yourself. So it's best to seek collaboration, even from a funding perspective, to actually move the needle.
So can you give an example of a good partnership that you've had where you've been able to optimize the data collection that you were keen on? At the same time, there was partnership between either an advocacy group or an external agency. If you can just share some light on what optimal partnership looks like for collecting patient data, that'll be helpful.
I can start, not necessarily for patient data, but for partnerships that have been successful in moving a project forward at different stages and that have led to kind of widespread collaboration within BCH and around the world. So one of the disorders that we work on is CDKL5 deficiency disorder. We initially created a cohort of lines from a foundation called Lulu Foundation. Those iPSC lines have been available to the research community around the world, and many people are using them to understand the disease better and to develop assays. As time went on, we realized in conversations with medical professionals, so Heather Elson leads the Center of Excellence here at Boston Children's Hospital, along with conversations with Tim Yu, identifying variants that are amenable to ASOs for exon skipping. We realized that there were patient variants not represented in the IPS lines we had available. So we partnered with another foundation, Cure5 Foundation, to now establish more lines that represent these other key areas that might be amenable to different therapeutic opportunities. Now we're collaborating with other labs at Children's, as well as other companies to develop assays to move drug screening platforms forward, as well as ASO development with different lines. Again, this came with funding from multiple different foundations. There's multiple companies now interested in using these lines, as well as academic labs. So that's one way that the seed of foundation funding can really expand to a large network of research.
And from a tool development perspective, instead of speaking about specific projects, I just widen the aperture a bit to talk about public-private partnerships. Critical Path Institute, other organizations that are nonprofit organizations, independent researchers. I've seen tremendous success in trying to actually develop clinical outcome assessments, partnering with technology providers to move the research forward to validate digital health technologies and digital endpoints. Pretty much cost sharing in all of these different tool development endeavors so that we can have something that's available to move the research forward. but I think it takes really thinking about the greater good to engage in these types of relationships to drive the tool development forward.
So if you can just share some insights as to how do you balance the need for speed to, kind of take a bus, not walk, but really, really jog along the path here of drug development. How do you see the optimal use of time can be, and what is the role of patients? I feel every turn you take, I think patients have a role in the process, and sometimes they know that, sometimes they don't know that. So if you can just kind of highlight your view of where the patient and the patient advocate fits into this journey and how they can help crunch the time to the process, I would really appreciate that.
I would say that just the context of the need for speed, I think everyone wants treatments to come to market sooner, and everyone wants a patient to have access to transformative therapy sooner. So everyone wants to try to set an objective to bring these drugs to market. However, there are a lot of trade-offs along the way. And I think patients and caregivers can really help providing input to precisely design studies in a way that they're measuring what matters most to them, tied back to the treatment target. The sooner that that can be realized, the better. And having the continual engagement of patients and caregivers throughout the medical product lifecycle also leads to a speedier time to market and potential for access for patients, because as you design studies at different phases throughout the development lifecycle, there are different trade-offs that have to be made. There are different things that you learn from previous studies and different refinements that you have to make to even the protocol for interventional and non-interventional studies. And that's where the input from patients and caregivers is very critical. That's where advocacy functions in sponsors or biotech companies and pharmaceutical companies like mine really play a part in cultivating that relationship. But I think along the way, that's where it's most critical to avoid delays, avoid setbacks, and allow for that strategic discussions on how best to measure what matters earliest, so that you can avoid, having to repeat studies or having data that's not usable. And can I also speak to the patient-led research, or do you want to?
Absolutely.
So I've been talking a lot about clinical trials, but there's a tremendous role that patient advocates, patients themselves have in generating evidence that can contribute to the medical product development journey. And that's through independent studies that can be designed through the organizations and serve as reliable evidence in the totality of evidence that's presented to regulators and to payers. We're thinking about like natural history studies, as well as registry studies, and others, even small independent research studies that serve as the basis for candidate selection. Even the littlest studies can often count if they're done well. And when it comes to decision making, so regulators, they're the ones to decide if it's approved. Payers decide if it's reimbursed and the pricing. And they want robust evidence, but they also know in rare disease, you're not going to get everything in one or two studies. So they're encouraging parallel studies, interventional studies in the real world to be designed and conducted. And that's where everyone here can play a part in driving this research forward. And it still is just as impactful as complementary evidence to clinical trials.
Thank you. I think that kind of brings a good closure to the discussion today. I think we've clearly heard that meaningful change to the landscape is emerging, but patients have a critical role to speed that up. Also, there is definitely a room and a spot for the patient at the table. The patient just needs to draw the chair and occupy that spot. And I think there is no resistance to that. There are enough advisors, there's enough regulatory guidance, and there is now the need for the community to connect the dots and collectively be on the journey for drug development together. As they say, if you want to go, slow, I think I got this wrong. If you want to go slow, if you want to reach your journey fast, you go alone. But if you want to be successful at it, you go with a team. So I think our choice here is obviously the latter. So I think what we fail to also acknowledge is the role for a group such as Global Genes that gives us the opportunity to bring everybody that is vested in drug development to be in one room, to network, to bounce off ideas, and to kind of find the common goal that we are all aspiring for, that is to bring therapies to market for rare disease patients. So with that, I will open it to the floor to ask questions to our panelists here. And if you have questions, please walk up to the speakers on each side of the room, and I can this one out.
I can also bring the mic if someone.
Well, while
Yeah.
people are thinking of questions, I can also maybe just address the idea of speed when developing preclinical tools and assay development, because oftentimes, I can feel the urgency from patient advocates, when we meet and they're, I want the stem cell lines made tomorrow, and it, takes a certain amount of time. And there's companies out there who will promise, a really fast turnaround time, but it's also important to pair with somebody who can ask the right question of, I've met with companies who are making stem cell lines, and I ask them, what are the karyotype of your starting material? And they tell me, and it's a little horrifying. And so, they don't see that as a problem, and they're not telling that openly unless you ask. So that's why it's really important to, yes, speed is important, but the quality of the starting material of these models is going to set up the entire phase for what you're bringing to the clinic. So to have the best success, take the time to make the right model with the right quality controls. And as Ebony said, there's standards for all of these things. So the International Society for Stem Cell Research has guidelines for best practices. Like, those really should be followed. If the groups you're working with aren't following them, ask them why, and really dig into those things. So that applies to all the model systems, but just wanted to put that plug too.
Hi, thank you both. This is probably for Ebony. First of all, congratulations and thanks. Could not agree more that there's a place for patient input early, can never be too early. The question goes more towards rare. Can you talk a little bit about the trade-offs of developing a new COA, a new PRO in a population that isn't well studied, that is so heterogeneous, that is just so scarce, and perhaps the sponsor company hasn't really undertaken a good diagnostic initiative?
Mm-hmm. Yeah, we run into that a lot, right? You can't have disease-specific measures for every rare disease or every condition for that matter. So there has to be some work that's done in order to see whether or not you can develop something that is applicable, and generate evidence early enough to understand what characterizes the condition, but it does take time. So in a situation where, you're moving really fast and you have very small sample sizes and you're trying to understand, well, how best do I measure this? It's not going to be perfect. You're not going to have a disease specific measure in a year. I've actually had that request before when I was at FDA, like, well, can we do that? I'm like, really? So you got to get creative. There are ways to take things off the shelf and to then do the qualitative work, which is a lower lift. And it's sort of good enough, not necessarily the best, but you can use qualitative research and quantitative research in parallel to determine what is actually best in a specific measure that may be off the shelf. But there are definitely trade-offs. When you have small sample sizes, even if you use the best measure, your trial may not be powered, or your study may not be powered to detect whether or not there's a treatment effect. That's where the totality of evidence comes in, I believe, looking at mixed methods approaches, drawing upon other kinds of conditions that are similar to drive concept coverage, determining which tools might best measure the most, impactful concepts, and then supplement that with qualitative insights from patients themselves. When you have a questionnaire and you're like, oh, is this result really true? But you also have an overwhelming amount of testimonials or qualitative interviews that show, oh no, this is actually changing in a patient's life, then all of that helps with interpretation. So I would say in rare disease, given that I've been doing it for so long, there's no silver bullet in one type of measurement approach. You have to have multiple studies on deck, and you have to have multiple ways to bring in the patient insights to interpret the quantitative results.
Yep.
Hi, Laura Budmeier from the ASXL Rare Research Endowment Foundation. Thank you for a great presentation. You talked about the importance of systematic and reliable data collection and like capturing how the patients like feel, function, survive. I think what's challenging sometimes is for a lot of the families who are on this journey, participating in all of these things, it's a lot of time, it's a big burden. Like, are you seeing any trends of, like you mentioned, wearables as an example, just like, are there other ways to reduce the friction to make this easy, to get more robust data from people, to help them to see more of the benefits along the way on the journey?
Yes, I'm seeing trends toward, I keep saying collaboration, but I think that is where the field is moving, pooling data, not actually stop talking to each other. I mean, hello, communicating about different studies that are ongoing within an organization. So let's talk about within an organization and across organizations. Within an organization, there has to be synergistic, strategic collaboration where we're mindful on the burden that is being placed on families, recognizing that we need to minimize our burden on them in these research activities, which includes how many times we're tapping them, so have a good thought ahead of time to say, okay, when we are trying to get them to input into this, how much time are they taking, et cetera. But then across organizations, those public-private partnerships that I talked about are critical. I think that there's more of a move toward trust, where people are willing to share data sets, to share insights, so that not everyone is doing the same thing at the same time over and over again and fatiguing, these families.
I guess from the biosample collection standpoint, just trying to, piggyback a research collection at the same time they're already present for a clinical appointment, so that it's not an extra day, another trip, another appointment to attend.
Question for Dr. Buttermore. Coming off of your story about the Lulu Foundation, I was curious, when a patient is the only person, or in the case of twins, the only two people with a specific mutation, and it's a disease that's maybe single digits in the world or in the country, how do you build a case for getting cell lines banked or getting biospecimens made? Is it just a matter of funding and finding someone to do it, or is it like the certain advocates?
Thanks. So I think if I understand the question, it's if it's very ultra rare, where is the funding coming from? So the Translational Neuroscience Center does support the collection of some patient samples as starting materials. So collecting blood and creating the PBMCs, which are starting materials for iPSCs. The larger funding for making the iPSCs is usually coming from a foundation in that case. But I think if there's a consensus mechanism and we understand that these variants might cause, both cause loss of function or something like that, there can be justification for making the cell lines, even if it's a ultra rare. Yeah.
Any more questions?
I got it.
Yeah, we'll take one more.
My name is Sumaya. I just founded the Dynamin One Epilepsy Foundation on the Rare Village. Dynamin One is just extremely, extremely devastating. And I have no words to say how awful the dystonia for these kids. They can spend hours really in extreme muscle spasms. My question is that for parents like us going through this terrible DE, developmental epileptic encephalopathy, which is predicted by Dr. Dennis Lal to be at the University of Texas to be at least over half of the incidence of Dravet. And it's a gain of function targetable. We have a program with NLORM. Thankfully, a parent started with them years before my daughter was there. The question here is that given that we have a straight path, how do we push for that or get that early fundraising to push for the registry, to push for the biobank? I mean, I think that's where the struggle is, that first push, because as parents, we are there. We're collaborating now with everyone in the UK. We have parents in China currently, and thanks to them, if they're hearing me, they're really funding their own programs there. We have multiple ways of targeting. We could just knock down the mutant and the kids can be saved. How can I push this fast? I'm based in Montreal and Toronto, so I'm working with sick kids to see if that program can actually match my daughter's mutation. But if there's anyone interested in Dynamin One, Professor Wayne Frankel was the professor who discovered the function in relation to epilepsy at Jackson Labs years ago. We worked with him as an advisor. Professor my cousin, thank you as well if he's listening as well, at the University of Edinburgh. We have a drug repurposing program that's starting there in his lab. If there's any way to help us push to just end that suffering, we have an Instagram by an amazing mother. The Instagram is Wilson the Warrior, and you would see how devastating this disease is. It's extremely stoppable. We could target it. There's no haploinsufficiency, but how could we get this fast enough to stop these patients from suffering? We have one patient just passed away. His name is Israel in Texas. I visited them last year, and it's more and more frustrating as a parent when you know you can. Like, I studied molecular biology. I worked in venture capital creation for a while. I'm a large cap, mid cap analyst as well. I think I could bring things together, but I'm just so lost on who could I latch onto, and I'm going to push as much as possible for this disease to be treated, because it has to be treated. And I'm sure a lot of patients here and parents echo this, to see children, they shouldn't be on wheelchairs. They shouldn't be suffering. Sorry.
No, yeah, thank you for sharing your story. It's so important to hear from patients and parents, and it reinvigorates the urgency of all of our work. I think you're already doing so many amazing things. The simultaneous movement of different platforms, from NLORM to drug repurposing, those are all the right things to be doing. Maybe we can have some more conversations with some of the clinicians who can provide more insight about, how to bridge those gaps to the clinic, because I think, again, it really goes back to having the partnerships and having team members on your crew that are from all the stages. So you're already, have programs, research programs developing, but bringing in the clinicians, the medical professionals into those discussions at this early stage as well. But happy to follow up. But thank you for sharing your story.
Any more questions? I think we'll wrap this session because I know there's a break coming up, but I just want to point out that this was just scratching the surface. Obviously, the roadmap is not linear, as you can see. It's a meandering path, and I think the next 2 days will hopefully provide the guidance we all are seeking to expedite that pathway. So please use the break times to network and connect and chat and find like-minded people amongst us so that we can all on this journey together in an expeditious manner. Thank you.